All right. All right. Today, our topic is coming to us from Dr. Paul Lawrence. He received his doctorate in microbiology from Stony Brook University. And before that, he obtained his bachelor's degree of science in molecular biology from the University of New Mexico. During his career, he has conducted biological research in academia, industry, and for the United States government. He has executed antiviral therapeutic and vaccine research at Brookhaven National Laboratory and the Plum Island Animal Disease Center. In industry, he has participated in contract biopharmaceutical projects for the former Collaborative BioAlliance and Dow Chemical. Dr. Lawrence was also the director of DNA production for the anti-counterfeiting biotechnology company Applied DNA Sciences. Currently, he is the executive director of research and discovery for Biocogent. In his scarce spare time, he is also the adjunct professor teaching microbiology to future nurses and doctors. Today's talk is going to be on non-coding RNAs and how they affect aged skin. All right, Paul, let's get started here.
Thank you very much for the introduction, Perry. All right, so let's get started. So this was the title of the poster that I delivered. It was an honor to deliver at the IFSCC meeting in London in 2022. And I'm going to not assume that you guys know anything about non-coding RNAs. This is the part of whenever I give a talk, I like to call it my brain calibration moment. But this is about epigenetics and epigenetic mechanisms. And this has been a popular topic on and off within the cosmetic care industry, and for good reason. But I don't want to assume that you guys know anything about epigenetics. So what does that mean? Epigenetics is basically the study of how we can toggle gene expression on and off without affecting any change to an individual's or an organism's genetic code. And there are three well-defined molecular mechanisms by which this can happen. There are actually four, but that one's relatively new, and I do not believe very germane to this conversation. But there's DNA methylation, and this happens in the nuclei of our cells where our genomic DNA is present, and sometimes sequences within our DNA can become methylated, and frequently this modification of the DNA basically turns off gene expression in that area, which means the activation of a gene and the production of a protein from the information encoded therein. Another mechanism that has been heavily studied is histone acetylation and deacetylation. So first of all, histones are these proteins that are found also in the nuclei of our cells that are cylindrical. And our DNA, because it's so extensively long, are compacted in the nuclei by winding it around these cylindrical molecules. And if the DNA is tightly wound around these proteins, gene expression is essentially turned off. But if we can loosen it, the gene expression apparatus can come in and begin to allow certain genes to be expressed. And there's two families of enzymes, one histone acetyltransferases, or HATs, that come in and will acetylate the histone proteins, which allows the DNA to be loosely wound. And then that's counterbalanced by a group of enzymes called histone deacetylases, or HDACs, that will come in and remove those acetyl groups and then restoring the tightly wound DNA. And that's how in the nucleus, another mechanism by which gene expression can be turned on and off, again, not affecting the genetic code. But there's this other one to the right, which is going to be the focus of my talk here today and was the focus of my poster in London. The discussion of endogenous microRNAs, and indeed a process known as RNA interference. And this is a process that happens mostly outside of the nucleus. So basically, if the prior two pathways didn't affect turning on or off the gene, the microRNAs can affect this change outside the nucleus of our cells. And I'm going to talk to you a little bit about this. So what happens is that there are these short non-coding RNAs. Now, before I get a little more into depth with these, I want to remind you of a little high school biology.
you.